Welcome, everybody. My name is Andrea Decker. I have the pleasure of working for the Fleet Science Center, and I'm very excited that you're joining us tonight for our Sharp Minds lecture series. Normally, this is a series in our beautiful Highcroft Dome Theater on the first Monday of each month. Unfortunately, we find ourselves in strange times, but that doesn't stop us of bringing science to you. And keeping you informed of what's going on in San Diego and on the important issues. And we are very, very excited that we can once again partner with the Sanford, Burnham, Prebys Medical Discovery Institute. They've been a wonderful partner for a couple of years now, and we look forward to continue working with them. And I'm very excited to have Dr. Pamela Itkin-Ansari with us today, who will give a wonderful presentation today on her research on diabetes.
Hello, I'm so glad to be here. I'm an Adjunct Associate Professor at Sanford, Burnham, Prebys Medical Discovery Institute, we call it SBP. And I earned my Ph.D. in Biomedical Sciences at UC San Diego, where I've been faculty as well. The first thing I want to tell you about is that the disease diabetes is primarily focused on the pancreas. 98% of the cells in the pancreas make digestive enzymes for us to digest our food, and those enzymes travel through duct cells into the gut. And it's only 2% of the pancreas. That's the islets containing the beta cells that make insulin. But it's insulin we can't live without. So the food, as it gets digested, much of it's converted to the sugar glucose. And glucose is really the body's fuel. The reason we need insulin is insulin's the key that unlocks gates in every one of our trillion cells in our body to allow the glucose to enter and be used as fuel. And what happens in type 1 diabetes is there's a shortage of insulin. So what you see in this figure are all the purple spheres are glucose being converted from our food in the gut. And those glucose molecules are winding up in the bloodstream, shown by the red cylinder. The problem is that there are very few green spheres, the insulin being made by the pancreas. And so that sugar stays in the bloodstream, and it causes problems when it stays in the bloodstream. And moreover, the cells in the body are starving because they don't have insulin to unlock the cell to allow the glucose to enter. So type 1 diabetes is an autoimmune disease. And the main problem is that the immune system essentially attacks itself. So in the image you're seeing on the left, the central part of this is an islet. And all the dark purple cells you see are immune cells that are infiltrating and attacking the islet. Now, type 2 diabetes begins very differently. And what this world map is showing you is a map of the presence of obesity in different regions of the world, starting with the least of the population that's obese in white, going through yellow, orange, red, and then finally to purple. Unfortunately, you can see that the United States is squarely in the purple zone, meaning more than 30% of our adult population is obese. And for reasons that are still being studied, what happens in type 2 diabetes is, at least initially, that the tissues in the body stop responding even when insulin's there. They don't open the gate and they don't take up the sugar. So if we use an image like the one we use to think about type 1 diabetes, now what you see in the bloodstream is a little different. We have all these glucose molecules being made by the gut, and those are the green spheres. And then the pancreas, at least at first, is actually making insulin, but the body isn't responding to the insulin and taking it up out of the bloodstream. And for that reason, it stays in the circulation where it causes trouble. And eventually, because the beta cell keeps getting a signal, there's high glucose, make more insulin, eventually this can lead to beta cell exhaustion, that the beta cell can't make more insulin correctly. And I'm going to tell you some of the specifics about that in a few minutes. But it actually winds up that 25% of patients with type 2 diabetes need insulin eventually because of this beta cell exhaustion. Well, one of the things that's really good to think about right now is we're coming upon the 100-year anniversary of the discovery of insulin. So 100 years ago today, it was uniformly lethal to have diabetes. Every child with type 1 diabetes died. And what you see in the image of the mother holding a child is a 7-year-old child that would eat, but his body was starving because there wasn't insulin in his body to tell the cells to take up the glucose for fuel. And right after insulin was discovered and administered to this little boy for 3 months, you can see the difference in the lower picture of how much healthier he looks. So discovering insulin is a miracle, and it's changed so many lives. So for type 1 diabetes, the main therapy at this moment is insulin injections or insulin pump. Now for type 2 diabetes, I mentioned that 25% of patients do need insulin, but we have a lot of other medications. And I show this slide just so you can see, you might see a medication you take or a family member takes and see what part of the body it's acting on. But a lot of what we're trying to accomplish is to make different organs in the body sensitive to insulin again. So they respond to insulin by taking up glucose. And so you can see we have drugs aimed at muscle or liver or gut. One story I particularly like about where some of these drugs came from really speaks to the need to do basic research and study our natural world, because we don't always know where great discoveries are going to come from. So where it says incretins under the insulin secretion and glucagon secretion and appetite control, one of those is called GLP-1, and it's a hormone we have in our bodies. And it's a wonderful hormone because it promotes beta cell function and beta cell health, and it does something else. It slows gastric emptying of the food. And why might that be important? The idea is not to overwhelm the body by producing all the glucose at one time if the body's going to have trouble utilizing it. Instead, if the food gets digested more slowly, then the body may be able to respond correctly and use that glucose better. Well, it turns out that we learned a lot from the Gila monster. This is a lizard that eats only 1 or 2 times a year, and it has a supercharged version of GLP-1 because when it eats a meal, it needs to digest that meal so slowly that it lasts for 6 months. And this knowledge and understanding their version of GLP-1 in the lizard has really helped us make some blockbuster, amazing drugs for type 2 diabetes. So we have insulin, and we have all these therapies people take for type 2 diabetes. Why do we need new therapies? Well, first I want to talk about the burden of insulin therapy. For patients with type 1 diabetes, and some of them were interviewed for a film the JDRF did in my lab a few years ago, and talking to children in the area with type 1, this little guy, Calvin, when he was 6, he'd already had 16,000 finger pricks to test his blood sugar and 10,000 insulin shots. That is not a carefree childhood that we all wish for our kids. And it really affects the entire family. I've had many parents tell me that since their child was diagnosed, they've never slept through the night. They get up in the night to check their child's blood glucose, to give them orange juice if it went too low, to give them a little bit of insulin if it went too high. So it's a full-time job for multiple people in the family. And years ago, a colleague of mine did a study to show what's causing the problems, even with all this effort to control blood sugar. The main reason we need new therapies is that we still have people getting complications of diabetes because we're not able to control the blood sugar well enough. So the graph I'm showing you is the blood sugar of a patient starting at 2 a.m. and going through till midnight the next day. And this patient is taking insulin shots to try and keep their blood sugar between the red bars, which is the normal region. But what you can see are these many excursions into hyperglycemia throughout the day. And this is despite a big effort. So what winds up happening is this chronically high blood sugar leads to heart disease and eye disease and kidney failure and peripheral arterial disease and amputations. And this is really tragic. And we don't have therapies that are keeping this under control well enough. And I lost my own father to diabetes when he was 63. This is one of the last pictures taken of him. He was showing my then 2-year-old daughter ducks in the water. And it's such a shame for everybody in our family that this happened. And I will be so gratified if in the end my work helps keep this from happening to other families. So I'm going to talk to you about 2 therapeutic approaches that we've been working on. One is a transplantation therapy, and the other is a way to improve beta cell function and survival in the insulin-producing cells that are retained in a diabetic patient. So it's important to know that patients with diabetes still have beta cells in their pancreas, but they're either not functioning correctly or they're very sick or on the verge of dying. So first I'm going to talk to you about transplantation. So this colleague of mine, James Shapiro, did a beautiful experiment years ago. He took that same patient who could not control their blood sugar with insulin shots, and he purified islets from people who had died from removing their pancreas, and he transplanted those islets into the portal vein. Actually, it was found the best place for the islets to lodge was actually around the liver. And some of those patients became insulin free. So it's very exciting, but it essentially remains an experimental procedure for 3 reasons. Number 1, there's a limited supply of islets. So we would never have enough islets to transplant all the patients that would need it. It isn't that often that a pancreas from someone who's passed away is in good enough shape that we can purify the islets. When you think of it, even if people have passed away, maybe from COVID-19, we would not take islets from a person who was infected to use as a transplant. The person has to have been ultimately quite healthy right up until they passed away. So we don't have enough supply. The second is that transporting those islets through the portal vein is an invasive procedure, not really amenable to something that could be done in a doctor's office, and carries some risks. The third and potentially most important reason that islet transplantation is limited is when one gets a transplant from another person, to keep the immune system from rejecting the transplant, the patient now needs to be immunosuppressed for the rest of their life. Well, that's not a great trade-off. If you think of 6-year-old Calvin, to potentially not have to take insulin shots but spent his whole life immunosuppressed and being worried about bad infections, we really need something better than that. So the goal in my lab was, would it be possible for us to develop a transplantation therapy that could use a virtually unlimited source of beta cells and would be minimally invasive and would not require immunosuppression in the patient? So that's a tall order. So first, what our group did is we showed that even cells that are developing in the body that are not fully beta cells can mature into insulin-producing beta cells after they're transplanted. And I want to show you all the work that's being done to convert stem cells into every type of tissue. The reason we're able to do this for beta cells, and that's the lineage shown in green in the middle, is because of 100 years of developmental biology research that people studied all of the tissues in the body and how they're formed from the primordial regions and what factors have to be expressed and what proteins have to be provided to signal for those factors to get expressed. In my lab and many, many other labs have contributed to our knowledge of how to get all these factors expressed. And what our lab did first was to show that cells that are in the transition, where I'll call them pre-beta cells, that they already carry the information, if we can get them to this point in the laboratory, that if we transplant them into an animal, they have the information to become fully functional insulin-producing cells. They don't need any other signals. In other words, they're destined at this point. And that turned out to be an important finding. Now I want to get to the issues of invasiveness of transplantation and the need for chronic immunosuppression. I investigated a technology called encapsulation. And basically the idea is, could we hide the insulin-producing cells from the immune system? So in this schematic, you see that we could load the insulin-producing cells from the port at the top. And they're shown in yellow with a purple center in the middle of this encapsulation device. And the device is made of a porous mesh. So this mesh has to have a pore size that's large enough that glucose from the bloodstream gets in to signal to the beta cells to secrete insulin out. So glucose has to get in and insulin has to get out. But then the pore size has to be small enough that immune cells can't invade. And so this is a very exciting idea because this is how we could potentially protect cells from the immune system. And in the image in black and white that you see is a picture of a device, an encapsulation device called the Theracite device that I used in my studies. And for studies in mice, the device was about the size of a small fingernail, and that could hold enough insulin-producing cells to cure the diabetes. So in the next slide, what I show you in the picture on the left is that after we transplanted these stem cell derived insulin producing pre-beta cells, they matured inside the device, and the arrows in that picture on the left are pointing to all the green insulin producing cells that formed after transplantation. And then the graph on the right is showing you that the green line says that a mouse that had diabetes that didn't get a transplant, that animal's glucose keeps rising and rising. But the red line across shows that if they got the transplant of the encapsulated cells, that we could control their blood sugar. And one added benefit of this approach is that the encapsulation device is, one, durable, and retrievable. So it could be removed if it ever needed to be. But most important, besides its ability to protect from the immune system, is we placed it just under the skin. And so not in any internal organs. And the insulin producing cells worked fine inside the device under the skin. Interestingly, for reasons we don't really understand, if we put these pre-beta cells under the skin without the encapsulation device, they don't do well. So this approach is now in clinical trials based on work from our lab and many other labs. And it'll be very exciting to see if this can be used on a large scale for patients with uncontrollable blood glucose levels. And now I want to tell you about a second approach we're taking, which is complementary. And that is, I mentioned that patients with diabetes still have beta cells in their pancreas, but they're not functioning correctly. Now, if we could get them to be healthier, that might stave off diabetes for some patients. But at the very least, what it would do is allow patients to take less medication. And for anyone with type 1 diabetes, you know that the worry long-term is complications from high blood glucose, but the short-term worry is always going low, taking too much insulin and becoming hypoglycemic. And if we could get some of the beta cells within the body to function better, we might be able to avoid needing so much insulin that the risk of going low blood sugar is considerable. So to get to this story of how we're working on the remaining insulin producing cells, I'm going to tell you how proteins are made. Proteins begin as beads on a string, and the beads are called amino acids, if you remember from school. And at first, this is linear, like the top left looks like an open necklace. And then the protein begins to fold. And importantly, for proteins to have a function in the body, they need to fold in a very precise manner. And the image on the right is a beautiful crystal structure of insulin. And you don't have to look at it long to be sure this is not random. This is a very carefully orchestrated folding and conformation that has to be achieved. And this is necessary for insulin to function. It turns out that protein folding is actually quite an energy-intensive job for the cell. And what we've realized over the years is how many diseases are, at least the part of the pathology is a problem in protein folding, and in some diseases, it's the entire problem. So I have teamed up with one of the investigators here at SBP, Randy Kaufman, whose life work has been dedicated to understanding how proteins fold. And what he and I are finding with our other collaborators is that diabetes belongs on this list of problems with folding. So let's talk a little about how proteins do get folded. Well, there are factors in the cell that are dedicated to this job, and they're called chaperones. And they exist in one area of the cell where the folding takes place. So they promote proper folding, and they prevent big globs of proteins from aggregating together and glomming up the works. And Randy and I like to describe how the chaperones work with a couple of these images from I Love Lucy. If you're my age, you might even remember this episode. So Lucy and Ethel are the chaperones working at a chocolate factory. And their job is to wrap and fold the paper around the chocolates coming down the conveyor belt. And when there's a reasonable amount of chocolate on the conveyor belt, they're doing a great job and all the papers get folded correctly. But as I mentioned, if the beta cell keeps getting a signal that there's too much glucose in the bloodstream, it keeps trying to make ever more insulin. And so what happens next is the conveyor belts speeding up, and the chaperones can't fold the proteins correctly. And so what you see here are unfolded products heading out. Eventually, this winds up in disaster. Either the cell dies, or it has to use massive amounts of energy to figure out how to get rid of all the unfolded protein. As you see here, Lucy and Elko are doing by putting them in their mouth or their clothes. So my lab set out to figure out in a human beta cell, what are the chaperones that are absolutely required for insulin to fold correctly? And so we did really what was a massive experiment where we purified all the precursor to insulin, because it's the precursor that gets folded. It's called proinsulin. And we purified all the proinsulin out of a sample of islets. And then we could precipitate that, and we sent it and all the proteins that were bound to it for analysis by something called mass spectrometry. And we identified the chaperones and another group of proteins that are required for folding. And I show you the main 20 here in work we recently published. And we've already begun determining which of these are the most critical. So we're very excited that we're well on our way to moving toward a therapy that would promote better folding of proinsulin so the cells don't become fatigued or die. One of my favorite things that's happened in research over the course of my career is how collaborative it's become. And so part of that is actually been made possible by new technology. So what I want to show you is that in our search for all the chaperones in the beta cell, we actually had to scan all the proteins that bound to proinsulin to determine if they were chaperones. And what this means is, although the experiment was expensive, it didn't cost any more money to define all of the hundreds of proteins that bind to proinsulin for different reasons in the beta cell at the same time that we were finding our chaperones. And so we're actually able to publish then a resource for the whole field. And this leads to new collaborations, people calling and saying that they're so excited about one of the interactors we found and would we be interested in looking at something with them. And it also, I benefit from these kinds of data sets that my colleagues then publish. They might have been interested in one specific area, but they happened to get so much information out of the experiment that now it's available for everybody in the world to use. So finally, we have identified a couple of chaperones that appear to be absolutely required for proinsulin to fold in human beta cells. Now, how do we take that information and help a patient? It's a process. But we at Sanford Burnham Prebys are in a very unique situation because we have a facility like what pharma companies have to do high throughput screening. And the center is called the Conrad Prebys Center for Chemical Genomics here. It was initially funded by a large grant from the National Institute of Health, and then made even better by a generous donation from Conrad Prebys. And so now we know a chaperone that we want to promote the activity of. And at Sanford Burnham in this center, we have about 500,000 chemicals, essentially a library of chemicals, just like a library of books, that we could test to see if any of them will enhance the activity of this chaperone. But how do we go about doing that? Well, there won't be a test on this, so don't worry. But these are images from our screening center. And I just want to give you some idea how all this works. So one of the first things we might do is to engineer the beta cells that we're going to use in a way that if they turn on the chaperone we're interested in, they'll actually light up. And to do this, we use a gene. Again, this came from just basic research into the natural world. We use the gene that allows fireflies to light up. It's called luciferase. Or we use a protein from jellyfish called green fluorescent protein. And so we've engineered these cells, and now we use robotics to dispense the same number of cells into thousands and thousands of little wells. We use plastic plates about the size of your hand, and each one has 1,500 little slots for cells. Now, after the cells are equally put in hundreds of plates with thousands of wells, then we dispense one chemical into each well. And to do this, the amounts are so minute that some of this is controlled by sound waves because we can't even measure it well another way. Then all these plates of cells are put in incubators, and each day the robots can remove the plates and put them in special microscopes that will scan for any cells that lit up. And then they'll send us the data. And from there, it's still quite a bit of work to see if the chemical really did this over and over again. And then we usually need to modify a chemical. Let's say it really is working. It needs modifications that our medicinal chemists do to make it stay for long periods in the bloodstream, or to get rid of any toxicity that could result from the chemical, or to make it more sensitive or more specific. So it can be years of work, but the most amazing thing is we can do it, and we can do it right here. So finally, I want to suggest that some of these approaches we're taking may not just be used when people are already sick, because we're at a point where we can predict who's going to get type 1 diabetes and type 2 diabetes to a great extent. In type 1, people develop antibodies against proteins, and these antibodies can be detected in a blood sample. And we know now that, for instance, if a child has antibodies to 1 or 2 or 3 proteins in their bloodstream, they're very likely to come down with type 1 diabetes in the next couple of years. So we'd really like to do some of these interventions early. And in terms of type 2 diabetes, what many of you may be familiar with is having your hemoglobin A1C or HbA1c tested at the doctor's office. What this is a measure when there's a lot of sugar, glucose staying in the bloodstream, it gets stuck places it doesn't belong. That's part of why there are complications to diabetes. One of the places it gets stuck is on this molecule. And the amount of sugar on the molecule is evidence of how high the blood sugar's been over the last 3 months or so. So when the hemoglobin A1C levels keep creeping up in people, this eventually gets to a stage we call pre-diabetes, where if the trajectory continues, they will have diabetes. And so we're really looking for ways to intervene now. Finally, I think you'll have learned something about Sanford Burnham Prebys tonight and about the work in my laboratory. So leaving with some knowledge. I hope you feel a little of the wonder of what's possible in science that I myself feel every day. And last, I want to leave you with hope that so many therapies are in the pipeline. The future is brighter for anybody who is dealing with diabetes right now. And then I'd like to thank my colleagues, the people in my lab who've done some of the work that I've discussed, and my colleague Randy Kaufman here at SVP, Peter Arvin and Ming Liu at the University of Michigan, and Bill Balch at the Scripps Research Institute. Thank you so much.
Thank you so much, Pamela. That was amazing. Actually, I learned so much, and it always baffles me that there's so much great work going on here in San Diego. So we have a question here. The percentage of people with type 2 diabetes, what is the correlation between overweight? I mean, is everybody with type 2 diabetes overweight, or?
The vast majority of people with type 2 diabetes are overweight. So, for instance, while type 1 diabetes is largely a genetically controlled disease, type 2 diabetes is a combination of genetics and lifestyle. So, there are people who are very thin and get type 2 diabetes for some reasons. We know a lot of genes that seem to play a small role in type 2 diabetes. But for the most part, what happens with obesity is fat tissue and other tissue start sending out inflammatory signals. And those signals interfere with the body taking up glucose and interfere with the beta cell making insulin. So even with type 1 diabetes, it's difficult to go back and not be diabetic anymore. But with type 2 diabetes, it's often found that if people lose a significant amount of weight and sometimes even if they exercise and didn't lose weight, that their diabetes gets well under control.
That's good to know. Thank you so much. Another question is, I don't know if you're familiar with it, but there is a distributed computer resource called Folding at Home
Oh,
that
okay.
people can participate in. And somebody's wondering if that has helped the research. I know of another program called Folded. If
Yeah.
people like to puzzle,
Yeah.
then they can go on Folded and they can help find ways to
Yeah.
fold proteins. Have you heard of these by any chance?
Well, yes, there are many programs to predict the structure of a protein. And in fact, there was just a fantastic advance in this technology that they're predicting protein structures with great accuracy. The reason that that's so important, so for instance, we know exactly what insulin looks like as when it's folded, and I showed you that picture. But to get that information, we had to turn it into a crystal and do what's called X-ray crystallography. Well, some proteins do not turn into crystals well. And so we don't know what their structure is. And then it's very difficult to potentially devise a drug that could activate or inactivate that protein. So if we can use these programs to devise the structure, we can figure out where the pockets are that we might want to design a drug to. So it is very exciting.
Oh, great. So I'm putting some links into the chat for everybody to see, because this way our audience, if they're interested in helping with the science and finding new structures, then this might be a way they can engage and help science out.
Yeah, yeah.
Find more answers. That's great.
Yeah. Like when people count the butterflies or the birds, or, the public can help in a lot of scientific research.
I love that. I love that we can have an active part in shaping
Yeah.
our future. I think that's wonderful. And then we have a question.
Uh-huh.
Encapsulated induced, and I hope I get this all right, encapsulated induced beta cells are not as responsive to regulatory signals that keep insulin matched to sugar levels. What are potential approaches to improve this regulation?
Yeah, so, I mentioned that our ability to make insulin producing beta cells is based on 100 years of studying development of the body, development of the pancreas. And we're still learning. So it's pretty amazing that we can make beta cells at all at this point, I think. And, part of the reason this has gone so well is that California funded the California Institute for Regenerative Medicine. And a lot of the work has taken place with funds from that institute, my own lab and other labs. But science probably still has a few secrets that we have yet to uncover that will improve the function of stem cell derived beta cells. I'll be very surprised if it takes us much longer because we've come so far.
And we've come so far in a relatively short time.
Yeah.
When you consider where we were 20 or 30 years ago, I think in the last
Right.
years, it's just grown exponentially once we were able to read the genome a little bit easier and
That's
cost
right.
effective than we were able to do. There's a question about type 1 diabetes. And why not just stop the autoimmune process? Apparently there is some work at the UT San Antonio using gene transfer with a viral vector. Is
Yeah.
that something that you can comment on?
Yeah, I would say half of diabetes scientists work on the immune system and how to halt the autoimmunity. And many, many things have been tried. But it is true that now that we have gene editing, we have CRISPR, it is much easy to manipulate cells in the body. And so progress has been made, but we haven't yet been able to turn off just the portion of the immune system that's misbehaving. Of course, we could shut down the whole immune system, but then the patient would be in that terrible situation of being immunosuppressed and prone to infections. So what we're learning, the more we learn about the immune system and the more we develop these tools for editing, we're able to hone in on the precise cell in the immune system that's causing the problem. And that's really the goal, to have precision in how you affect the immune system.
And does that tie into, there's another question about the research at Mass General Hospital using Bacillus calmette guerin BCG vaccine to prevent and reverse autoimmune diseases? Is that...
So there are vaccines, there are multiple vaccines being tested right now. And I think they do hold promise. And so what's so exciting, and I think the reason that diabetic patients should be so optimistic, is that there are all these avenues being tested. And one reason we want to take all these shots on goal isn't just because we don't know which ones will fail, but because in the end, we may need personalized medicine. It may depend in part on what your genes are, which one of these therapies will work best to control your immune system if you have type 1 diabetes.
That makes a lot of sense. Everything seems to be so personalized.
Yeah.
And this seems to be a good way to go. Another question that we have is, I expect your research to go tremendously well and you'll figure this out, but what would be the next step to get that into clinical trial and into patients' therapy? How does that work?
Okay, so if we talk about the transplantation therapy first, that's already been through all of the animal studies, and it's in patients now in San Diego and around the country, I think even in other places in the world. So that's already in clinical trials. If we talk more about how a discovery made in my lab, for instance, finding a particular chaperone that's necessary for pro-insulin folding, how do we get that to become a medication for a patient? That's quite a process that begins with the high throughput screening facility that I showed you that we have. So we come up with a lead chemical. And then we need to do a lot of studies in cells in the lab to make sure that chemical is behaving correctly. And ultimately, we have to do some studies in animals to make sure it behaves correctly before we go into patients.
And that obviously takes time.
It takes time, and it takes quite a bit of money.
Yeah, I could imagine. Everything that takes time takes a lot of money. There was another question. Oh, I know a lot of people with COVID-19 are quite concerned about getting sick, and there was the warning that people with diabetes are at a higher risk. Can you explain why that is?
Yeah, so with type 1 diabetes, of course, it's based on the immune system going awry, right? It's an autoimmune disease. But what we understand now about type 2 diabetes is that these tissues that when there's obesity, they're sending out signals like from fat tissue called adipokines, and some of these cause a lot of inflammation in the body. I would say low grade inflammation that's always there. It's kind of like having a slight fever, the way I picture it. And so if you now add a serious disease on top of that, like COVID-19, then the patient was already at a slight disadvantage, already having inflammatory problems, and now you're adding a major problem on top of it. And that's one reason why people with diabetes will be in a group likely before the general public to get the vaccine to protect them.
That makes a lot of sense. Thank you for that. And we have another question. How do you purify proinsulin for use?
Well, proinsulin we don't give to patients, but we give them insulin, right? But I'll answer two things in the case I misunderstood the question. The way we purify proinsulin to do our studies, to find all the proteins that are interacting with it, for instance, is we use an antibody that binds specifically to proinsulin and doesn't bind to insulin. And my colleague Bill Balch made that antibody. It was quite an effort to make one that would be that specific. But maybe the question is more about how do we make insulin for patients. And because we now know the sequence of the gene, we can create as much insulin in the laboratory as needed. You know, initially people used to get insulin that was purified from cows or pigs. But now we have the human sequence and we have ways to produce massive amounts. So there's no shortage of insulin.
Wonderful, thank you. Another question we have is, since we know that type 2 diabetes is related with weight and obesity, there is something called metmorphin that is said to help people lose weight. Is that true? And if so, how does it work?
So metformin is a really interesting drug. It acts on a lot of different tissues. And in some patients, it does cause them to lose weight, not in everyone. And it can often control diabetes even when weight hasn't changed. But it has many different functions. So it's really a very special drug in the arsenal for type 2 diabetes.
Thank you. And I've got one more question. I have heard a lot about gene therapies, especially for autoimmune people with autoimmune diseases. So they take out the bone marrow, they do some gene editing on it, and then transplant it back in to change the production of cells, just like beta cells. Would that be something foreseeable for the future as a solution?
Okay, so for type 1 diabetes, bone marrow transplants have been tried. And so the idea here is that you take out some immune cells, but then you purge the patient's body of all of the rest of their immune cells. And what you put back are either the patient's own or other cells to repopulate the immune system so they won't have autoimmune disease. The issue with bone marrow transplantation is that it's very high risk. The patient is in quite a bit of danger at the time when they're purged of all of their own immune system. And it hasn't actually even worked for all the patients that have been in the trials. But the gene therapy idea, I think we're really coming very close to that. It's related to the issue of being able to edit genes, in that we're finding some, the pinpointing the immune cells that we might want to change so that we either attack those or we alter them. Sometimes it's possible to get a cell type to differentiate into a different cell type. So can we get the autoreactive T cells, for instance, to continue on a course of differentiation into something else, into a related cell. So these are all approaches that are being studied. And as I said, probably half of the diabetes scientists are working on the immune aspects of diabetes.
I'm glad that there's so many different approaches to
Yeah.
tackle that problem, right? Because it seems like there could be multiple solutions and different angles,
Exactly.
especially since type 1 and type 2 are different.
You know, what's important is we call it the etiology. How they begin is different, but ultimately they both result in too much sugar in the bloodstream, and that's what leads to the complications. So the complications in type 1 and type 2 are exactly the same because it's caused by that high blood sugar. And ultimately, in both diseases, the insulin producing cells fail to keep up with demand and then become damaged or die. So in that sense, the pathology begins differently, but the end result is very similar.
It's very similar. That is a good segue to the next question that we have. We have a attendee here, a guest here, who was misdiagnosed as type 2 diabetes initially at the age of 48. And then he demanded some testing and had to start insulin therapy for the past 4 years to be treated as type 1 diabetes. And he was wondering, is childhood diabetes always sudden?
Is it always what?
Sudden.
Sudden. No, but you know, you hear very dramatic cases where suddenly a child might pass out and be taken to the hospital and their blood sugar is hugely high. But it's not always sudden. Sometimes somebody with diabetes will start being thirstier than normal, fatigued. And so if someone's looking for these symptoms, they may be able to recognize it. And I've known quite a few people like this attendee who were initially diagnosed with type 2 diabetes. You know, when you think about the Juvenile Diabetes Research Foundation, the other name for type 1 diabetes was juvenile diabetes, because I would say 95% of the cases occur in children. And so sometimes it's a little overlooked that it could be type 1 diabetes in an adult, but eventually it gets figured out.
But it could start, type 1 diabetes could start later in your life. It doesn't have to start.
It absolutely can. It's just that I would say in 95% of the cases, it begins in children. And we used to consider type 2 a disease of adults. But sadly, because the obesity epidemic is also happening in our children, we're seeing quite a lot of type 2 diabetes in children, which is not, you know, that's really a shame, because then that's a burden they're going to have for a long, long time, as opposed to someone getting type 2 diabetes maybe when they're 65 or 70.
Yeah, I agree. But the good news is that we have so many wonderful scientists working on a solution.
We do, and we understand, you know, lifestyle changes we can all make that none of us want to make.
Yeah, it's hard. I
It's
like my
hard.
donuts once in a while.
Yeah.
And especially the holidays are coming up, that's not going to make it easy.
Oh, and being stuck, you know, in isolation with COVID-19, I don't think this is helping anyone eat better and exercise more. So, but it's good to think about, because people can make a big change in their health on their own.
I think that's a good note to end on. We
Great.
should all take a closer look at keeping us healthy, keeping us fit, and keeping us well. I want to thank you, Pamela, for this wonderful talk. I really appreciate all the information and you answering all the questions.
Yes, happy to do it. I was happy to be here. Thank you.
Thank you. And before we say goodbye, I quickly want to point out, for anybody who had missed the beginning of our talk, because we have another partnership with the Sanford Burnham Prebys Medical Discovery Institute coming up. It's a 5-part series on the science of aging. So we will be looking at the problems that we might encounter as we age. So the series is called Live Longer, Live Stronger, and it features all the work that SBP is doing to help us age in a healthier way. In January, it's always on the first Wednesday of each month in the afternoon from 2 to 3. And in January, we'll be looking at healthy aging to prevent cancer. And in February, we'll look at some Alzheimer's disease risk factors. In March, we're going to learn how the heart ages. In April, we will look at preventing vision loss as we age. And in May, we're going to take a look at our bones and how we can stay strong as we age, which will probably talk a little bit about exercise. So here we are again, talking about exercise, eating right, and keeping us healthy, especially in these very trying times. Again, thank you so much.
Okay, thank you,
Pamela, and thank you everybody for joining us. And I hope we'll see you again for the upcoming series in January. Everybody have a safe and wonderful holiday season. Goodbye.
Bye-bye.